I have no power. All right, here is your micro trivia question. One of your classmates actually sent me this article. I had seen the headline before the child died, but the child ended up dying of what organism? So these are all common causes of meningitis, and I gave you one from each group. Neisseria meningitidis is one of the most common causes of fatal bacterial meningitis. West Nile virus, we have all over the country, is a very common viral cause of meningitis. Cryptococcus neoformans is the most common cause of fungal meningitis, and it's found in bird feces, so anywhere birds poop. And Naegleria fowleri is the most common, or one of the more common eukaryotic causes of meningitis, or amoebic causes of meningitis. So it's a parasite. Fungi are eukaryotes too. Sometimes people call Naegleria fowleri the brain-eating amoeba. So which one of these fairly common causes of meningitis led to the death of a child near Dallas? I'll give you 20 seconds to talk about it. It's like a zombie amoeba, like a brain-eating one. All right, who thinks bacterial Neisseria meningitidis? A couple people. West Nile. Okay. Cryptococcus neoformans. Okay, and the brain eater. Okay. Brain-eating amoeba, Naegleria fowleri. This is one of those infections that in the entire United States, throughout history of tracking this infection, only 4 people have ever survived. One person had a very avirulent strain. It hardly caused any tissue damage in cell culture. The other 3 people who survived were children. They were diagnosed and treated immediately. Treatment involves anti-amoebal drugs and therapeutic hypothermia. You have to lower the body temperature enough so that the brain doesn't swell and cause death. One of the children who most recently survived back in 2016, he was at a hospital in Orlando, and the drug happened to be manufactured in a facility right down the street. So he was able to get the drug right away. In 2013 was the time we had 2 children survive. One child was treated fairly quickly and made a full recovery. The other was treated with some delay and has significant permanent brain damage. So this is a very, very nasty infection. You find it very commonly in places that are warm during the summer. So this amoeba is highly associated with like swimming holes, canals, right? Pools that are not chlorinated very well. And it just goes right up your nose and burrows into your brain. So it's a pretty gnarly infection. And so this is a headline we see every year in this country because of this amoeba. Okay, so we left off on Monday talking about how organisms grow, and I showed you that classic bacterial growth curve. We talked about binary fission. And then I told you that one of the key features that can affect growth is temperature. And I showed you this graph over here, where if the temperature is too low, the membrane will kind of gel up, and it prevents exchange of nutrients. It prevents cell function because you can't get stuff in, you can't get stuff out. As we increase temperature, protein function gets better. Proteins also slow down when it gets cold. And so every protein within a cell will have an optimum temperature. It's usually the temperature of the body. You don't have too many proteins that function at a different temperature from your body. But very, very quickly, we can go over the maximum temperature, and we can permanently destroy those proteins. And I told you that there are some proteins called chaperones. When we talked about protein folding, chaperones can protect from heat damage. So E. coli has a huge family of proteins called heat shock proteins that protect them from damage. But as I'm sure most of you know, once you heat up proteins, they never go back to their normal shape. And so the example that we always talk about are eggs, right? You open up an egg, it's all liquidy. You heat it up, it's all solid. You can put it back in the fridge and it doesn't get re-liquidy. You have changed the shape of the proteins. You have denatured the proteins irreversibly. So once this happens, that's often why you can get cell death at higher temperatures. The proteins just cannot tolerate the high temperatures, they denature, and the cell can die. You can also have collapse of the cytoplasmic membrane. Remember, your cytoplasmic membrane is full of things like fats, and what happens when you heat up fats? They go from solid to liquid, right? You have a nice big steak, and you can see all those white pieces of fat. You put it on the grill, and that fat liquefies. Well, that's what's happening to your cell membrane. Right? It liquefies and falls apart. And of course, your cells are mostly water. What happens when you heat up water? It boils. So the cells can explode from the boiling of the cytoplasm. Being heated to death is not a nice way to go. Okay. So when we talk about things like us and most animals, we tend to fall in a very narrow range of temperature that we can survive. And we would normally classify ourselves as mesophiles. We survive at what's considered to be a mid-range temperature. And most of the microorganisms that any of you who are interested in medicine are going to be interested in are also mesophiles. Because if you're interested in medicine, you're interested in human health and things that can affect human health. And of course, those are going to be microorganisms that grow at human body temperature. But we have a wide range of microorganisms that survive in areas that are not normal human body temperature. And so we can classify organisms based on the temperatures that are optimum for them using these four categories, excuse me, I can't count this morning. 4. Psychrophiles. Psychrophiles like the cold. Their optimum temperature is significantly lower than human body temperature. These are microorganisms that might do very well in the Arctic or Antarctic. They can drill into glaciers and find living microorganisms. I don't know if you, on social media, if you follow like some of our national parks, Yosemite National Park posted this really cool image a few months ago of these microorganisms that grow on the snow and turn the snow pink because of the microorganisms. We don't grow in the snow, right? We have to maintain our warm body temperature. If you tried to just live outside in the snow, you would die pretty quickly. But microorganisms can do very well in some of those cold environments. There are also some microorganisms that cause human disease that can do well in cold environments. An example is Listeria monocytogenes. It's a very, not a very common cause of foodborne illness, but when it does cause foodborne illness, it's a common cause of death. So it's a really dangerous foodborne infection. And it not only can survive being refrigerated, it actually will grow and replicate inside of your refrigerator. So some of the recent outbreaks of Listeria involved things like cantaloupe. Because what's your first, you get a cantaloupe, I love cantaloupe, right? You get a cantaloupe, you're gonna eat it, so you take your big knife, you cut into the cantaloupe, and then you store the rest in the fridge. But how many of you actually like scrub the outside of your cantaloupe before you cut it open? Not very many, one hand. So all of the bacteria that were on the outside of your cantaloupe get dragged right in as you cut it open. And Listeria grows in the refrigerator, right? That's why you put things in the fridge, to keep them from spoiling. Well, Listeria doesn't care. The other food product where we had an outbreak of Listeria was ice cream. And I get really emotional about it. Before I moved here, I lived in Texas. And it's hot in Texas if you've ever been to Texas. It's humid in Texas. And sometimes the only thing that makes you feel better is ice cream. And the summer that I was going to move out here, the summer of 2015, there was an outbreak of listeria in the Bluebell factories. And Bluebell, I'm sorry to say, even though I grew up in California, Bluebell is the best ice cream around, and it's Texas ice cream. It's delicious. It's also full of listeria, it turns out. And they found the outbreak because 3 patients at a hospital in Kansas were given a little small thing of Bluebell ice cream as part of their hospital meal, and they died. Right? Like, ice cream can kill you. So this is a microorganism that thrives in those cooler temperatures, but also thrives inside of your body. So it has a very wide range of temperatures it will tolerate. And that is not super common. For the most part, when we think about bacteria that cause human disease, if you put it in the fridge, you stop the growth. You put it in the freezer, you probably kill them. But listeria is just like, oh, that's nice. Oh, it's cold? I like the cold. Oh, it's hot? I like the heat too. Great. Okay, but that's kind of rare. And then we can talk about microorganisms that like hot temperatures. And we call them thermophiles. So thermophiles will be in areas where the temperature is consistently well above what we would consider to be normal, like well above human body temperature. And then we have our hyperthermophiles. And our hyperthermophiles are those that are found in places like deep sea thermal vents, your hot springs, like at Yellowstone. So there are microorganisms that are able to not only survive, but thrive at various temperatures around the planet. And sometimes we've been able to take advantage of those organisms. If you ever do go to Yellowstone, the enzyme that we use to do PCR, Taq polymerase, is isolated from a microorganism called Thermus aquaticus that was taken from a deep sea thermal vent. Or not a deep sea thermal vent, one of the hot springs, right, that they have there at Yellowstone. And because its optimum temperature is really, really high, you can run a PCR between like 50 and 90 something degrees C, and the enzyme doesn't degrade because that's where the organism lives. So when we have these organisms that exist in these more extreme environments, we call them extremophiles. They love extreme environments, very hot or very cold. We can also talk about very acidic or very alkaline. So any environment basically that we can't tolerate, we think it's extreme. And we say that they are extremophiles. And so what they're showing you here are some examples of cold places where they have isolated microorganisms. Okay, so glaciers, ice cores, etc. And then here is the snow algae. So this is a picture of what I was telling you about. Here's the snow, and then this is the microorganism. It's an algae that can grow on the snow and turns it pink. So I was going to do that as a micro trivia question, but it ended up coming up today before I used it. But it's an algae that turns the snow pink. And so the National Park staff at Yosemite put it on Facebook and Instagram. So thought that that was really cool. So again, just because we don't tolerate a temperature doesn't mean that there isn't a thriving community that maybe we can't see. Sometimes we can. But there are going to be thousands and thousands of microorganisms in these environments that we think are intolerable. Okay, so how do we live in the cold? Because I told you we can't live in the cold. So how do we, the royal we, the microorganisms, survive when it's cold? I told you when it gets cold, our membranes gel up, our proteins stop working. Remember, the inside of our cell is mostly water. When water gets cold, it freezes. So how do microbes prevent those things from happening when they're living inside of a glacier, in an ice core, in that really cold water? Well, one of the first things that they can do is alter the way that they structure their proteins. And organisms that have evolved to tolerate the cold generally have incorporated more alpha helices into their proteins. If you picture an alpha helix kind of like a spring, they're more bendy and they have more flexibility. So when it gets cold, they won't stiffen up and stop working. When you have a lot of beta sheets, you're more likely to be more rigid and less flexible. So they incorporate flexibility through the use of alpha helices. They also use more polar and fewer hydrophobic amino acids. And they don't incorporate very many weak bonds. They use some stronger bonds. So they modify their proteins in such a way that they can have that flexibility with the alpha helices, that they can not get rigid and fracture when it's cold, right? That's sometimes what things do when they get cold. They get so rigid and so brittle that they'll break. And so by incorporating strong bonds, you can prevent that breaking as well, because strong bonds are harder to break. What else can microorganisms do to tolerate the cold? So I've told you a little bit about how they can alter their proteins. Well, they can also alter their membrane. So organisms that are found in cold environments generally have high levels of unsaturated fatty acids in their membranes, and they're usually a little bit shorter. So if you picture a fatty acid, picture your phospholipid bilayer, and we always kind of illustrate it having like a little Z pattern. And so when you have that little Z pattern, you can imagine it's fairly easy to pack that really tightly together. And the reason that we have that Z pattern is when we have a saturated fatty acid, one that's full of hydrogens. So you would have something, I'm just going to draw one layer, your fatty acid. Right, you put another one. Right, they'll pack pretty tightly together. But when you start to incorporate unsaturated fatty acids, you start to put double bonds into the tails. And when you start to put double bonds into some of these tails, you introduce new kinks that make it harder to pack together. So you could have a double bond here, maybe there's a double bond here, right? A double bond here, something like that. So by removing some of the hydrogens, by having these unsaturated fatty acids, you can delay the gelling. It's harder to get it to come together because it's just not going to pack as tightly. They'll even have some that are polyunsaturated, so multiple areas on the same fatty acid tail that will not have enough hydrogens. So you can have multiple double bonded carbons, which can make the tail even more bent. So they can adjust their membranes in that way. I told you that one of the things that cells can do when they get too hot is express these chaperone proteins that help prevent protein misfolding. They can also do that in the cold. So there's a family of proteins that are called cold shock proteins. They're induced when the organism gets colder than optimum. And that can, again, help with protein folding. So it can help with a process to keep proteins being synthesized and to keep them being folded. And it can help if proteins begin to denature because of the cold. And some organisms adapted to the cold even bring their own antifreeze. So some organisms that are cold adapted make cryoprotectants. So antifreeze proteins, certain solutes, different sugars or sugar alcohols that prevent freezing of the cytoplasm. So if you ever find yourself working in a molecular biology lab or a microbiology lab, we store bacteria for use later, and we put them in a -80, so -80 degrees. That's really, really cold. I actually burned my hand cleaning out the -80 last week. Well, how do I keep my cells alive? If I just take a culture of cells and put them at -80, they're all going to die, because I usually work with things that cause human disease. They want to be at 37. I'm like, no, -80. Well, we add a cryoprotectant. We add something like glycerol or DMSO. And what that does is it prevents the cells from forming ice crystals so they don't explode and die. And when I'm ready, maybe in 6 months or a year or in 10 years, when I am a full professor and can do what I want, I can go back and wake up my cells. So we know about these types of things. We know that we can preserve our cells in the -80 because we know that organisms can survive at very, very low temperatures. And it's not just microbes that have these properties. I went to a really cool talk a few years ago from a student working in one of the chemistry labs that they were looking at fish, that live in very, very, very cold environments that also have antifreeze proteins circulating throughout their blood. So it's a fairly common adaptive strategy to tolerate the cold. And of course, we can form a biofilm or a slime layer or a capsule. And that almost acts like a little blanket of goo to protect you from getting too cold. Try to bring that home. Can I have a goo blanket, please? Keep me warm.
Did you find any of those antifreeze proteins in eukaryotes?
Yeah, like in fish. Yeah. There are some fish that have different types of cryoprotectants.
Like that?
Most mammals, no. Most mammals just don't tolerate that cold of temperatures. Yeah, I mean, so even things like we think about, there are mammals that live in those environments, right? Like in Alaska, there's bears and moose and caribou and all that stuff. They usually have a lot of fur and they have a lot of fat to keep their temperature warm. They can't go down to that cold temperature. They have to continue to be warm. But things like fish that don't regulate their body temperature in the same way, they'd have to have some sort of protective mechanism. So there are some eukaryotic multicellular animals that can do at least the cryoprotectant. Yeah. Okay. So any questions about how microbes can survive the cold? I don't think we have to worry about that too much anymore. The whole planet's just going to burn up anyway. So these cold ones, they're like, well, I did okay during the Ice Age, but these guys are going to be the ones that outlive us. Our thermophiles, those microorganisms that can survive at warmer temperatures. So above 65, so 65 is kind of this cutoff where we only see prokaryotes. So as far as we are aware, eukaryotic organisms have a thermal maximum, and it's approximately 65 degrees. For you, you're going to be dead anyway at 65 degrees. But there are some eukaryotes, some fungi, some algae that can tolerate those higher temperatures. So fish and other aquatic vertebrates, their maximum is usually 38, so just above our body temperature. Insects can go a little bit higher, and so can crustaceans, maybe up to 50. Most vascular plants have an upper limit of 45, though one species has been clocked at 60. And mosses, again, about 50. So you can see that for the most part, eukaryotes, 65 is really hot. But after that, it's all microbial life. So there are some bacteria that can go all the way up to 95. so just below boiling. And then there are some archaea at 122, which is hotter than boiling. So these microorganisms can tolerate much higher temperatures than eukaryotic cells can. Oops. And so if we talk about life in a hot spring, some of our hyperthermophiles, we have both chemoorganotrophs and chemolithotrophs. We have archaea and bacteria. We have a much higher diversity of life than you would think. And the generation time can be as low as 1 hour. So these can be rapidly growing microorganisms that, again, not just surviving high temperatures or tolerating high temperatures, but actually thrive in these really high temperatures. And so here they're going to show you a picture of a hot spring and then kind of a zoom in of all of the different cells that were found in that hot spring. So it may not be as much life as we see at some of the more mesophilic temperatures, but still the abundance and diversity of life at these extreme conditions was really surprising for scientists to discover. So our thermophiles, if we call an organism a thermophile, we're talking about one that lives in a moderately hot or intermittently hot environment. So it's maybe not consistently super hot, but it can get pretty hot and those organisms can survive. And what we generally see when we have an environment that's really, really hot is we can actually see a thermal gradient. So if we talk about like a deep sea thermal vent, we all think of the bottom of the ocean as being very, very cold. And that, for the most part, is accurate. It is quite cold at the bottom of the ocean. But you have these deep sea thermal vents that can be hundreds and hundreds of degrees right at the thermal vent. And as you get farther and farther away, as you might imagine, the temperature begins to get lower and lower and lower. And so you see different abundance and different diversity of life at all of those different gradients. And so their distribution then is dictated by their biology. If you can tolerate the really, really, really high temperatures, you'll be as close to that thermal vent as possible. If you can tolerate kind of middle of the road high temperatures, you might be farther back. And if you're a psychrophile, you're like, ooh, thermal vent, no thanks. I'm going to be over here in this cold area. And so you can actually see the gradient and see the diversity of life change. And so here they're trying to show you again kind of an area in a hot spring and then how their enzymes function. And so this is just some enzyme from an organism isolated from a hot spring, and they're calculating enzymatic activity at different temperatures. And so at 90 degrees C, this enzyme has 100% functionality. It works really, really well. Even at 100, we increase the temperature to boiling. There is a decrease in activity, but not much. Now, when we get up to 110, we can, get rid of all the activity. So we've reached that area inside of that cell that would be their thermal maximum, somewhere between 100 and 110. However, by adding something like calcium or another cofactor that can improve enzymatic function, we still have enzymatic function even at 110. It's not peak, it's not optimal, but the enzymes can still work. So these are what we call heat-stable enzymes. And that's why we've been able to take advantage of some of these organisms and use them in the laboratory for things like PCR. Because we run PCR at really high temperatures. We don't run it over 100. The highest temperature that we do in my lab during PCR is there's a cycle at 95, somewhere down around 60, and then back up again at 72. Peak function at 90-something. So we have enzymes that function very, very well at these high temperatures that we've been able to take advantage of. And so what we'll finish off with today is how do microorganisms tolerate those high temperatures? I told you when it gets too hot, you basically, your membrane falls apart, you boil, and all your proteins get denatured. You turn into an egg, right? You are boiled, you're hard boiled, and there's no going back from that. So what cells that thrive at these very high temperatures have are evolutionary adaptations that make life easy for them at those temperatures. They substitute out their amino acids just like the cold-tolerant organisms do. But in this case, they put in amino acids that help provide stability in the heat. So shapes that are less likely to denature under hot conditions. They have increased numbers of ionic bonds that are less likely to unfold as the cytoplasm heats up, right, as we boil. They have really hydrophobic interiors, so they exclude that hot water or hot liquid from getting inside the middle of those proteins, so they can protect the temperature inside their proteins. And again, just like cold-tolerant microorganisms can produce cryoprotective compounds, heat-tolerant organisms can produce heat protection compounds. So they can produce solutes that help stabilize the temperature. Just because water boils at 100 degrees doesn't mean other liquids boil at 100 degrees. And so if you have different solutes inside of those liquids, you can alter the boiling point and prevent thermal lysis. And of course, their enzymes are commercially useful, like we've talked about with the Taq polymerase. And they also modify their cytoplasmic membrane. So instead of having these unsaturated fatty acids like cold-tolerant microbes do, they have lots of saturated fatty acids so that the membrane is pretty tightly packed together. And when it warms up and things start moving around a little bit because we've heated up, it's less likely to fall apart because it was already really tightly packed. And archaea have a really cool adaptation. Some of the archaea that live at really high temperatures, instead of having a lipid bilayer, have a lipid monolayer. So a classic lipid bilayer is going to be, like this. But our archaeal monolayer is going to look like that. And what happens when you get too hot and you have a bilayer is the two layers will begin to separate. If you only have one layer, it's not going to separate. So that ability to have a monolayer could partially explain why some archaea are able to survive at even higher temperatures than our bacteria. We'll pick up with acids on Friday. I gave all of your exams to the laboratory instructors, so they will be handing back your exams. I do not have your exams, the laboratory instructors do. So get your exams from them.